Okay, exam 3. It's going to be Thursday. Right, so coming up to the end, right? So it's going to cover chapter 20.12 through 23.9. This is the amides. All right, so we didn't cover amides on the last test, so that goes all the way back to carboxylic acids. And then this gets us to our organometallics. Which we covered last Tuesday, right? In the organometallics, that section is the metathesis. This covers all of our enolate chemistry, right? So remember, anytime you're doing an enolate, focus on creating the enolate. And then reactor enolate. So a majority of this is going to be chapter 21, which is really enolates. I'd say that's the hard part of this. Amines, we've done some of those reactions. There's a few specific amine reactions you should focus on. The organometallics, there's really a couple of classes on there, and most of them are named reactions. They're pretty straightforward reactions, not going to get tricky. The ones that are going to probably catch you and was on the quiz last week, especially people do well on the quiz last week, was enolates, right? So I would focus on those. Don't ignore the other chapters, but the enolates is going to be the part that's going to be tough on this one. Okay, any questions about the exam? We also have quiz 4. Right. Quiz 4 is going to be a review quiz, right? Because of the guidelines with the last week of the semester, this is going to have to be next Tuesday. Okay, so it's going to cover everything we covered this semester. Okay, kind of get you back up to speed. Okay, again, 5 questions through the semester. And then our final exam is just a couple of weeks from now. Final exam is December 14th. Right. Now the final exam is somewhat comprehensive. There'll be a little bit more off the last sections. The last sections is really going to be today and what we can cover on Tuesday. The last day of class, we'll do a review and kind of work some problems through the whole semester to get you ready for the final exams. But at this point, really, we're only dealing with new content today, a little bit of new content on Tuesday, and then getting ready for the final. And you can think of quiz 4 as a way to get you started on getting ready for the final, okay? And all of these are going to be online. If you want to come to class, you can. But if you want to take it where you've been taking your quizzes, fine. Same strategies as far as the way I've been setting the exam. I'm going to keep doing those. I'm trust you on the academic integrity. So moving forward, we got carbohydrates and proteins are really the 2 sections we need to cover now. Both of these are more to get you ready for biochemistry. Many of you are going to have to take biochemistry. For those of you that don't have to take biochemistry, this is also to get you prepared for some of the discussions when you get to advanced biology classes. So carbohydrates really are what we can think of as sugars. If we take carbon dioxide and water, right, and do some photosynthesis with these, this is what we end up with, right, forming our sugars. Right, so we're going to end up with this. The way to think about this is that the formula C6H12O6, right? That's where it gets the carbon, right? And hydrate, water. So the chemical formula of our base carbohydrates are always going to be the same. It's going to be C1H2O1, right? Some combination of that. Your base structures for carbohydrates are actually going to be aldehydes, as we can see this one, right? Okay, so this would be an aldose. Dose here, the OSE, right? That's going to be the sugar telling me I have a carbohydrate. Oh, added too many. Right, and then these are going to be ketose. And again, the OS tells you the ketone, gives you the ketose. Now, one, because I'm not a biochemist, and two, because it's a lot of drawing. I'm going to pull up a lot of pictures today. I'll give you enough time to draw them out. They're also in your book. Photo library here. Okay, so if we look at our kind of fundamental aldoses and ketoses, we have glucose and fructose. So this is an aldose. All right, this is a ketose. Right, both of them have 6 carbons, right? So both of them are C6H12O6. And they're both shown in a Fisher drawing. Okay. Now in organic 1, you might have covered Fishers, you might not have. Fishers is a way of demonstrating stereochemistry. So whenever it has it to the left and the right, what a Fisher is doing is when I see something like this, whatever is going up and down is going back. So that means this aldehyde group and the CH2O2 is going back. And then whatever is horizontal is coming at you. So although it just looks like crosses and hashes, it's actually showing the stereochemistry. Now, Fisher projections make a lot of sense when you're dealing with carbohydrates, but organic chemists tend to not like them. One of the reasons we don't like them is if we think about this glucose molecule up here, if I turn it on its side, what I'm actually saying now is, let me go up. So let's put our aldehyde here. The way this is drawn, it's drawn in a eclipsed form. And so if we look at this, if we took it around to the side, Okay, so the drawing here is in an eclipsed formation, right? So therefore, that's one of the reasons that we use it, right? But since fissures are eclipsed, then what would be the highest energy conformation? Eclipsed, right? So a fissure by definition is always going to show the least likely conformation of any molecule. But it makes it easy, as we see when we get to more sugars, because it's easy. You can say right, right, left, right, and that's glucose, right? So you can learn this pattern real quick. The other thing that we get to these is that it shows you the stereochemistry pretty quickly. And so let's look at, bring up a few more and look at the next ones. Okay, and that's what's shown here. Now, whenever we have all of these, you notice they're D. Okay. All sugars, all natural sugars are D. So all of these, anytime you have a stereocenter, right, we usually have the possibility of an R and an S, right? And if we had equal mixtures of an R and an S, then that would be racemic. All natural sugars, all are D sugars. You can produce L sugars, but no natural processes create L sugars, right? And so the fact that this is on that side would be the D sugar. That defines the D sugar. If we looked at an L sugar, right, this molecule would be L-ribose. So it'd be the mirror image of the D-ribose. So it's really this last carbon down here that determines whether something is L or D. It is the arrangement of the hydroxides, right, that tell me whether it's a ribose, a aeroribose, a xylose, or a lysose. So you can think about it, ribose, they're all on the same side. Aeroribose, we got the first one to the left and the other two to the right. Xylose, we go right, left, right, and then we go left, left, right, right? When you get to biochemistry, not in here, but when you get to biochemistry, you will need to know these, right? We should be able to know these at site. And so the Fisher projections are really helpful with that because you can get down, you can make your own little code on how to learn those. When we get to amino acids, you'll see that our amino acids are similar. There's only one set. Okay, those were all of our 5 carbon sugars. Right. So these are our pentose. Right. So pent, 5, ose, sugar. So these are all of our 5 sugars. The ribose, you should be familiar with that, right? We think about RNA, that's a ribose. So the R in RNA is for ribose. If we look at these are our 6 carbon sugars, right? So hexose, 6. Again, all D, these are the natural ones. Allose, altrose, glucose, right? That's an important one. Mannose, you'll see that one. Glucose, if you're, you'll see that one. Galactose, if you are lactose intolerant. Galactose is the sugar in lactose that makes that. We'll talk about lactose in a little bit. So, and you can see here again, it's the D is based on the blue bottom, right? They're all to the right. That makes it D, but then the arrangement of my OHs determines what kind of sugar I have. And again, I'm not going to require you to know this, but you will. It's usually one of the first things you learn in biochem. And it's not hard, especially if you use the Fisher projections. One of the other aspects of this is if we think about the synthesis of sugars, they start with just carbon dioxide, right? But then as they add carbon dioxide, we're adding one carbon. And so we actually can go in this kind of family tree. The first molecule we make that we would think of as a sugar is actually this D-glyceraldehyde, right? So the D-glyceraldehyde is our first kind of proto sugar. And then as we add carbons, we can end up with the 4 carbons, right? For our tetrose, 4 carbons, then our row of pentose, right? So ribose, adenose, xylose, lexose. And then from there, right, there's families. So ribose actually will create the allose and the maltose. The arabinose creates glucose and mannose. Then our xylose creates a glucose and idose. And then easy for me to say, right? D-lysose and then galactose and talose, right? So there's a class of these molecules. And again, that's another way that you can learn these. So we think about it, you start with CO2, you add a carbon, add a carbon, add a carbon. What you'll learn in biochem too is whenever you take those carbons to turn sugars into energy, right? We all know about sugars energy. You're also clipping them off one carbon at a time, right? So you're going to take a glucose and turn it into a ribose, right? And then a ribose is going to be turned into ethers, which will become a glyceraldehyde, which will then be turned into CO2 and formaldehyde. And these charts are in your book. So if you don't have time to write them down, you can look them up. You can also just look them up and then if we look at our ketoses, so this is our family for aldose, right? Notice these are all aldehydes. Here are our ketoses. And let's get a bigger page. Blow those up again. Right, and so we can see here that we start with the dihydroxyl acetone, right? That's a symmetric molecule. Then that turns into my etherylose, right? And then add one more carbon, get down to ribulose and xylulose, then pisose, fructose, sorbose, and tagose, right? And then this is the one that we're going to be seeing quite often, fructose. All right, so fructose and glucose is what makes sucrose. We'll look at that today as well. I think that's all the pictures for right now. Yeah. Okay. So these are all of the sugars that we're having and the names. The things to take away from this is really understanding your geometries, right? The D and the L. So the D is natural. The things like pentose, hectose. The other things to think about are your aldose and ketoses. And then, and that's the base structures that we need to be concerned with. The other thing that can happen with these is remember that if we take a aldehyde or a ketone, right, when we have an alcohol and the aldehyde, if we add you know, a little bit of catalyst to that, a little bit of acid catalyst, I can turn that into a hemiacetal. And we looked at this, right? There's an alcohol. And we looked at this when we were going through hemiacetals, ketals, acetals. But if you notice over here, I've shown it going up, but I could have also shown it with the OH going down. And that's because when this cyclizes, I can either take the OH from the back, right? Or the OH could have come right from the front. And then that will give me whether that hemiacetal, right, the stereocenter around that hemiacetal, whether it's going to be facing up, right, or, well, yeah, facing up or down. Oops. So if I take glucose, so if I take glucose, this can cyclize. It'll take a little time. So this is a Hawthorne projection. Okay, so what I'm showing here is not a 3D projection. It's a specific projection that's used kind of like Fisher, just for sugars. Okay, and now I have the OH up. We're going to call this version the beta. And then do the same thing on the bottom. Oops, I just redrew it. Okay, and this is going to be my alpha. The full name for the first one would be beta D glucopyranose. Now the pyranose part of this name is for the 6-membered ring. So you can have a furanose, which would be a 5-membered ring, or you can have a pyranose, which would be a 6-membered ring. So glucose can make a 5-membered furanose. So you could have beta D glucopyranose, right? Or you can have the pyranose. This is the pyranose. So pyranose is 6-membered rings, furanose is 5-membered rings. Yes. Yeah, that's actually going to be this double bond. Right. So if we look at starting here, 1, 2, 3, 4, 5, 6. It's going to be 1, 2, 3, 4, 5, 6. So the aldehyde oxygen is this oxygen, right? So if it attacks from one face, right, then it's going to end up going up. If it attacks from the other face, it's going to end up going down. So the aldehyde position is a position that gives me the alpha or the beta. Now, if I had, let's say, the oxygen on the 3 attack instead of the oxygen on the 2, then that would have given me a 5-membered ring. Now, hexanoses tend to make pyranoses, but they can make furanoses. Pentanoses tend to make furanoses, right? But they can't make pyranoses, right? So you have a little bit of there, but in most cases, if you see the pyranose, what they're just telling you, don't get confused by that. It's just telling you that it's a cyclized ring versus a straight chain. Now, the alpha and the beta are important. And they're important because when we get to larger structures, it's going to actually change what that structure does. So if we think about sugars, the sugars, we don't think about them always being the mono sugar. So what we looked at those charts, right, these were all single sugars. We're going to start putting the sugars together. And at that point, these alphas and these betas will make a huge difference in reactivity. And that'll be important to remember. So if we look at this is the Hawthorne. Now let's go ahead and convert these into the chair formation, right? So we're used to seeing this type of formation, right? So let's draw these two in this formation and see if we can figure out what the difference is. That's not obvious when we look at the Hawthorne projection. Okay, so if we have the glucose here, we have the OH going up. So this is going to be the beta, hydrogen. Then in the equatorial position, I'm going to put the OH. And now I'm only going to show the OHs, not the hydrogens, except for my alpha. And that's kind of done for classical reasons. Okay, so here's my beta glucose. And then over here, we have our OH going down, this OH, that OH, this OH, CH2, OH there. Okay, and this is my alpha. So that's my alpha D glucose, or alpha D glucopyranose. Now, the important things to think about here is if we look at each of these positions, right, what is that position? When we think about chair conformations, what is the important, remember all the way back to chair conformations, right? What was the important distinction, whether it was equatorial or axial, right? So we had axial and equatorial. So here what we need to think about is what position these are in. So if we look at my CH2OH, that's in my equatorial, right? My OH here is in the equatorial, this OH is in the equatorial, this OH is in the equatorial, and that OH is in the equatorial. So when we look at the beta D gluco, right? Everything on the beta is in the equatorial position. When we look at the alpha, again, we have equatorial, 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 but what now, where's my OH at that alpha position? Well, it's axial, right? So between these two, which one is more stable? The beta is more stable, right? The beta is more stable because it's in the equatorial position. The axial is going to be less stable. The other thing that we're going to be thinking about is if I was going to substitute, let's say that I had these two positions here. Now let's put a tosylate on here, right? If we were going to add a nucleophile, which of these two is going to accept the nucleophile more easily? I should move this nucleophile up here. Well, this one's coming in the equatorial area, right? So therefore, less steric hindrance. This one's coming from the axial position, so therefore more steric hindrance. So the alpha is less stable, but the alpha is also what? More reactive to backside attack, right? So that's going to be important as we start to look at our disaccharides and our polysaccharides. Now we're going to spend a lot of time on reactions of carbohydrates, but it's important to know a few things, right? One is that these are aldehydes, right? So I can take and oxidize these up to acids, right? So the way to think about the reactivity of carbohydrates is to think about the fact that they are at their root aldehydes, alcohols, and ketones. So aldehydes, ketones, and alcohols. I can also take these with reducing conditions. And in this case, it actually is something as simple as lithium, not lithium aluminum, that will catch on fire, sodium borohydride, right, in alcohol, and that would reduce this down to the alcohol. And I'm not showing all of the structure here. There's a few other reactions that aren't going to be on the learning objectives that's in the chapter. Just focus on the ones that you've seen before. So you've seen reductions of aldehydes. I just showed one, right? And you've seen oxidations of aldehydes. There's some other ones that are shown in there on how to increase the length of a sugar, how to make a glucoside, different things like that. Ones to focus on are, these are aldehydes and these are alcohols, and they will do reactions of aldehydes and alcohols. That's what I want you to focus on. Okay. And again, I'll put some learning outcomes up, and that'll hopefully specify what you need to look for. Okay, disaccharides. So the di here is going to be 2. So we're going to be taking 2 sugars. So one of the ones that used to be my favorite before I needed to cut back on gluten, since it doesn't like me anymore, was this one. Okay, this is maltose. So malt sugar, right? So if you take barley and other grains and you start to germinate those and then stop the process before they fully germinate, you'll create malt, right? Malt has malt sugar in it, maltose. Okay, now let's look at this disaccharide and kind of break it down. Based on what's going on here, can you tell me what sugar that is? Where are all of my groups that are attached? Are they all in the equatorial position except for that one? Yes. That's glucose, right? So this is a glucose molecule. And if we look over here, again, they're all in the same position. This is glucose. And then what type of attachment do I have here? Is this a beta or an alpha from this position? Is it going up or is it going down? It's going down, right? This is going to be an alpha. So we can put maltose as, we can show it as alpha 1,4 glucose, sometimes diglucose, sometimes. If I spell it right, it'll make it a lot easier. So the alpha is the type of linkage. And that comes from the alpha and beta that we saw previously, how they cyclize. So this has got an alpha, and then the 1,4 is how it's attached. So if we look at this position, 1, 2, 3, 4, and over here, if we go 1, 2, 3, 4, right, we've got a 1,4 attachment. Your common attachments are 1,4 or 1,6. So maltose is a 1,4 alpha linkage between two glucose molecules. Okay, that is the disaccharide. We look at sucrose and start out a little ways because I'm going to actually draw that direction. This one is sucrose. Again, if we break down this molecule over here, that's on its side, so it might be hard to recognize, but notice that all the attachments are in the equatorial position. This is glucose. But then this one over here, we haven't seen this one before. This is fructose. This is a 5-membered ketose, right? Or that's my ketone 6-member sugar, my fructose. So sucrose is a glucose and a fructose coming together. Again, looking at your glucose up here, right? This is an alpha linkage. And it turns out that the linkage is still a 1,4. Questions about these? Let's look at a couple of others. Again, these are in your book, so if you don't have time to draw it. Here, this is cellubiose, right? So cellulose. But notice here, right, in this middle, that now this is a beta. So our position right here, this is an alpha, this is beta, right? If we compare that, and again, this is a glycoside, so it's 2 glucose molecules. If we compare that to the one above, which was the beta, the maltose, right? What's the big difference between the two? Well, one of them is got the beta and the other one's got the alpha. Now think about where you might have heard these before, right? Maltose, malt sugar, use malt sugar to make beer. You put malt into malts to make them not shakes, but malts, right? They're sweet, the little candies, that's for malt, right? Covered with chocolate. But what about cellulose or cellubiose? Have you heard of that? Would you eat cellulose? Yeah, we eat it all the time. But what does it do for you? You can't digest it, right? So it's fiber, right? So fiber, cellulose, it's plant material, right? You can't digest it. The reason you can't digest it is the beta, right? So this position right here, right, is hard to hydrolyze, right? Because of that steric hindrance. Since that's hard to hydrolyze, you can't break this down, right? Insects have bacteria with special, so termites, right, have special bacteria that break this bond, right? But we don't have that. So this bond is not breakable, but this bond is. So this is easily hydrolyzed. When it hydrolyzes, it turns into glucose. It gets absorbed into the blood system. If we don't have enough energy, we use it up right away. If we have energy, then it gets stored, and it gets stored as a starch so that we can get back later, right? And then if you get too much starch, right, then all of a sudden it starts to get stored by a more long pathway, which is fat cells, right? So it gets processed into fat, and then the fat is even a longer storage. So starch, fat. Okay, cellubiose. And the last one that we'll look at specifically for this one is the lactose, right? So another one that you'll see a lot of is lactose. The reason that some people have problems with lactose is the galactose molecule, right? So you don't have the enzyme to break this down, right? Again, this is a beta. You have a higher energy species. Notice that this is axial, right? But you can't break this apart, right? So therefore, it can cause you digestive troubles. Lactose intolerance is not the same thing as a milk allergy, right? So I've got a nephew that's got a milk allergy. That's different than my wife's lactose intolerance, right? So it also turns out that people with lactose intolerance usually can have cheese. Why? Process of making cheese. What does the bacteria do to the milk? Well, it digests that and it turns it into the lactose into regular sugars, right? So it eats them up. So it breaks all the lactose down. So unless it's really fresh sugar, cheese, you've broken all the lactose down into its subcompounds, right? And in most cases, you don't think of cheese as being sweet because the sugar has already been consumed. Okay. Right, questions about disaccharides? Yes. Yes, right. And so your beta linkages are going to be harder to hydrolyze because of the steric hindrance between the two. So they're more stable, right? So they're lower energy. And then they have a steric hindrance to getting a nucleophile attack, where the alpha is a higher energy, right? And so, and I've got an open side for attack. So I've got an advantage to hydrolyze alpha linkages over beta linkages. Yeah, so, and we'll see that when we get into polysaccharides next. And the same reason. Okay, questions? Okay. Again, these are in your book, so don't feel like you have to copy them all down. So the cellubiose becomes cellulose, right? And we can see here that when we get into cellulose, right, we're just continuing it out. So when you get the polysaccharides, that's just going to be long chains of these, right? And again, all of these are beta linkages. If we look at amylose, which is a starch, here we've got all alpha linkages. And I forgot to draw to grab the picture of the amylopectin. So if we think about amylose as being kind of linear, because all the connections are 1,4, when we get to amylopectin, what we have is we have branching. Because you have a lot of 1,4, which is kind of linear, but every once in a while you'll have a 1,6, which causes a branch, right? So you start to get a little branch. So the main difference, both of these are starches. You can think about amylopectins as being starches that are a little bit harder to metabolize, takes a little bit longer to get them out, where amylose starches are ones that are a little bit easier to metabolize because of the branching. If you think about amylose, once you start a chain, you just run the whole thing down, right? You just start hydrolyzing, and you clip off those sugars left and right. When you get to amylopectin, you start running a chain and then you hit a branch. And so you got to start over, right? So it just slows down the process of doing those. Okay. Questions about these polysaccharides? I think that was the only ones I had pulled out to show you. Okay. So let's take a look at RNA and DNA. Wait, is that what I want to do? Yeah. Oh. Again, these are in your book. But the important part for DNA is this deoxy. So if we look at the base structure, right, we've got a nucleotide that's attached to this deoxyribose, right? And what we're missing is that OH. So if we look at ribose in this form, we have this extra OH. And the deoxy, we lost that OH. Now, why that's important is it turns out that without that extra OH on there, the deoxy, this is more stable. So the base structures of these are very similar, right? There's a little bit of difference in nucleotides. We'll take a look at that. But their base connectivity is going to be based on a ribose. But the deoxyribose, without that extra OH there, makes this a more stable molecule. With the OH there, all of a sudden I can hydrolyze that, meaning that that's easier to break down. And if you think about the structures, DNA is the base pattern, right? That's the base information. think about that as being in hardwired, where the RNA is going to be soft. I'm going to create the RNA from the DNA, but do I want the RNA to stick around all the time? No, I want it to go away, right? So the RNA is only there for a minimum amount of time, where the DNA, I want it there all the time. And so this difference of this OH, right, is the difference between the stability of these two structures. The way they work, other than that, is very, very similar. What you'd want to, if you want to pattern them off of each other, right? And of course, when we look at the structure, it's not just the ribose, it's also this phosphate, right? So we've got the phosphate molecule here. We have our ribose, our deoxyribose in this case, our phosphate chain, and then our nucleotides, right? So our DNA chain is made from these phosphates and the deoxy. And without the extra OH here, this is going to make this chain more stable. Now, the way this works, I think, is also important to cover. And again, these pictures are in your book, but I didn't want to take the time to draw them. If we think about what's going on in the messaging, right? So if we think about DNA providing information, how do we provide information in electronic means, digital? We have what? Ones and zeros, right? So all digital information, everything you're seeing on a computer, right? It's all based on a binary, either a 1 or a 0, right? All the data is coming through. But what is DNA then set up on? It's not a binary system, it's a quaternary system, right? And that what is the basis of that quaternary system? Not on and off, but what? 3 hydrogen bonds, right? And 2 hydrogen bonds. So the base pairs, right? The cytosine and the guanine, they have 3 hydrogen bonds, and then the adenine and thymine have 2 hydrogen bonds. Okay. And so, and we can put them backwards, right? So we have 4, we have a whole system based on 4. So instead of on and off 2, right? We have A, B, C, D, or C, G, A, T, okay, to get those. Now, if we were looking at these 2, and you were looking at a sequence in DNA or RNA, but specifically DNA, how would you know where you could start or stop? What would be a sequence clue that you were going to start or stop? Now, think about these things being zipped together, right? Have you ever had a zipper break? Right, really frustrating, right? You'd want it to be at the beginning or the end, right? So you don't want it to come apart in the middle, because then you're just kind of frustrated, right? So what type of bonds do I want at the beginning and end of an important sequence? I'd want places where it's easy for me to open up that zipper. AT. So what you see is you see sequences of A and T together. So you see ATTA, TATA, ATAT, right? You see sequences usually in 4s, but you see sequences of AT. That usually tells you you're starting a marker. So when you think about all the stuff they've done with genetics and using CRISPR to take this code out of that fish to put it, so all of a sudden you have glow in the dark fish or glow in the dark shrimp, right? What they're doing is they're using the weakness of the 2 hydrogen bonds versus the 3 hydrogen bonds. The thing that I find really interesting is, from a chemical standpoint, DNA and everything in life is based off the information you get off of DNA, is based on a couple of fundamental, very first semester general chemistry, organic chemistry, which is you got 3 hydrogen bonds versus 2 hydrogen bonds. And by missing an OH, you have a stable structure versus one you can reuse all the time, right? And so your base structures for this is very important. Now, one of the things I think it's important to go ahead and cover, I'm going to talk about because it's come up and I've had to explain it. The mRNA vaccine, right, is made from the standpoint that mRNA, messenger RNA, but it's RNA, right, is based on a information that's gotten from the DNA. So think about the process. I have the DNA makes the RNA, right? But the thing that RNA never does is do what? Make DNA. What makes DNA? Other DNA, right? DNA replicates itself. So when you have an mRNA, one of the things that was out there that the mRNA vaccines could change your DNA. That's not plausible, right? What can change your DNA? Mistakes. Where do you get mistakes? You get mistakes usually from radicals. How can you get radicals? Well, burning compounds into your lungs to make free radicals, like smoking, being out in the sun too much and getting light to create that, putting things in your body that tend to make those, or things that kind of look like cytosine and kind of look like guanine that can get put into your body. And all of a sudden it's like, oh, wait, that's not right. I thought you were a guanine, but you're not a guanine. And I put you in here, but now you're not quite a guanine, you're not quite an adenine, and I'm going to make a mistake. So DNA changes come from mistakes in the DNA code, but RNA is not part of that process, right? And so when you're thinking about the mRNA vaccines, the mRNA vaccines can be made very quickly because all I need is the DNA code of the virus. That I can then use to make the RNA to create the right protein. And once I've identified the protein, and then the RNA I send into your body, it goes into the mechanism that creates protein, and now your body creates a protein. The way other vaccines work is that you take the virus, you break it down so it's not viral, right? And then you put that in there and it's already got the protein because it was a live virus at some point, right? And so, you know, one of the things that's really come up is, why did they do that so fast? Well, because they've been researching mRNA and how to do this for decades. And second is, you cannot change DNA from an RNA, right? And so that's not the way the process works. Let's pull up the RNA structure so you can see the differences. And again, the base difference is the ribose sugar versus the diribose, and then a little bit of difference in our base pair, right? We don't use the thymine, we use the uracil. And again, because of this extra OH right here, that allows it to hydrolyze and break apart faster. So I can break the RNA back into its parts. So if you think about it, life is all based on a really, really beautiful, beautiful, beautiful Lego set. So, you know, if you're a Lego person, you'll love bioengineering. It's fun. Okay, questions about that? Yes, ma'am. Yeah, ingesting things are from the environment, right? So if you think about the cancer caused by, let's say, Agent Orange, one of the compounds in Agent Orange had a aromatic nitrogen containing species. And then once through the environment, right, from being exposed to Agent Orange, those soldiers, both sides, got exposed to that. And if that soaked into their skin, it could replace the cytosine or the adenine, right, base pair and get put into there. And then when it tried to replicate, it wasn't replicating it right. So that's one thing. Another one is they used to use certain chemicals. It's a misnomer. There is a chemical that you'll find in chemistry labs that's similar, but it's not the same molecule, dioxin. But dioxin, a true dioxin is just a 6-member ring with 2 oxygens on it. But that oxen they're talking about is an oxygen-containing aromatic that came from fracturing tar, and they use that on, let's say, light post, used to use this on light post, right? And you could be exposed to that. And that could then be, because the OHs aren't that different reactivity as the NHs, you would see that going. So what happens is you get some of these from exposures, right? You can get these, there's some compounds like the sodium nitrite. What happens with sodium nitrite, if you consume too much of it, it tends to make free radicals, right? And then the free radicals can methylate. And what happens is all of a sudden, if you notice, let me see, is it up here? What I can do is I can add a methyl to the nitrogen. All of a sudden, it looks like a 2 bonded instead of a 3 bonded. So if we look right here, right, one of the problems you can have is methylation. And if I methylate right here, then all of a sudden it starts behaving like it has 2 hydrogen bonds instead of 3, right? So there's several things. Most of them are environmental, digested, right, type things, right? Now there's also just mistakes that can happen, right? It's replicating this all the time, and you can get mistakes. There's also genetically related mistakes, meaning your DNA code has a weak linkage maybe somewhere where you can get a, so there are, that's what when they say, you know, you can get tested for breast cancer or something like that. There's some markers in there that could easily flip. So, okay, other questions? And of course I did synthesis on nitrogen containing aromatics. So who knows, you know, but I'm really, knock on Formica. I'm glad I don't have anything yet. So it's one of the reasons PPE is very important when you're doing research, because you don't ever know what you're working on. Okay, do we have a little time left? Yeah. Okay, I want to just introduce amino acids, and then we'll finish up amino acids and proteins next time. The idea of an amino acid is just a carboxylic acid with an amine on it, right? So it's just this basic structure. And then of course we have all these different R's. So if we think about it, DNA can be transposed into RNA, right? So that's the code that makes the RNA, and then the RNA can be used to make proteins. And the proteins then can be used to make enzymes and other structures, right? And the enzymes are our catalysts that get other reactions to go, right? So we're driving down. So we go from the 4 to now the amino acid pairs. And this will be big, and again, it's in your book, so don't try and copy it. But when we look at our amino acids, and again, you'll have to learn all these when you get to biochem, or I've heard molecular biology. You've got your amino acids, right? And we start with the small ones, but all of these and the next chart as well are going to be the ones that are used to make up, well, everything. So they're the building blocks for everything. Now there's some classes of these to think about. Try and make this bigger. So these first column here are just standard alkyls, right? So we go from hydrogen to the methyl, to the isopropyl, to this isobutyl. Then we have the sec-butyl, right? And then we have some that, you know, have different, but it's just in our group with a thiol. This is a pyrroline. Then a couple of phenyl ones, right? The tryptophan has got the nitrogen on there. And then here now we're getting into ones that are have polar side groups. So we have amides, set of amides. We got alcohols, right? We got the phenol, and then we have the cysteine, which is the sulfur. And then we have some that are acidic, right? So these would be groups that have an acid side chain. And then we have the group that have a base side chain. So and when we're looking at these different groups, right, are going to cause different conformational changes, right? So if we look at a hydrogen, we don't have much conformational change, but when we have something like this, right, that's going to cause it to spin. If we look at something like the phenylalanine or the tryptophan, now I have this kind of big wing out there. I have this big flat molecule that works kind of like this big blocker. But then whenever I have things like this and that, now all of a sudden with the acids, I got a protonation source. So if I think about an enzyme with a reactivity site, those acids, whenever I see something with an acid on it, that could be a reactivity site. When I see something with a nitrogen on it, right, that's going to be a proton sink. So like pyridine, where we pull the proton off, that could be a reactivity site. So when I'm looking at the protein structures, I can use these to kind of figure out what potentially is going on. Same thing up here with the amides. Then the cysteine, this one is really important. Remember when we looked at our disulfide bonds and we talked about, you know, curly hair and straight hair and how you could relax hair or you could perm hair, right? That has to do with the amount of cysteines and the oxidation going from a thiol, right, to a disulfide, right? So what happens with these is if you have a lot of these, you can form covalent bonds between proteins, where in most cases, right, with these, you're forming hydrogen bonds, right? To make amino acids, see if I can get one more page. You're going to give me a page, you're just going to let me know. Yeah, there you go. So synthesis. Some of these are review. This first one is, if I take an acid, I can use bromine and PBr3, followed by water. Okay, and this is the Heck-Selinsky reaction. And then all I need to react is take that bromide and now add an excess of amine. Now, the problem with both of these is both of them are racemic. And notice on all of my amino acids here, all the ones that have a stereocenter all have the nitrogen coming out at us, right? So they're all L amino acids. So all natural occurring amino acids, right, are going to be L amino acids. So all natural occurring sugars are going to be D sugars. So the only problem with this synthesis is I come up with the racemic mixture, and I would have to figure out a way to separate the racemic mixture. We'll stop there. We got a couple more reactions to go.